Dear students, today let us start lesson on cellular division and cell cycle. Cell division is the process by which a parent cell divides into two or more daughter cells. There are different stages of mitosis altogether define the mitotic phase of an animal cell cycle. This division of the mother cell into two genetically identical daughter cells is nothing but cell division. Fleming, who is from Germany, was the first man to document the details of the cellular division. There are different kinds of cell division as follows. Let me start. Number 1, amitosis. It is also called karyostenosis or direct cell division or binary fission. It is a cell proliferation that does not follow mitosis. The polyploid macronucleus found in some ciliates divides amitotically. This type of cell division starts by the simple cleavage of the nucleus and the division of the cytoplasm without formation of spindle fiber and appearance of chromosome. This is also called as direct cell division, commonly seen in simple organisms like prokaryotes. This picture tells about amitosis, where the cleavage starts in both nucleus and cytoplasm, and then there is a formation of a two daughter cell without involvement of chromosome or spindle fiber. Amitosis is the method of direct cell division in which the nucleus divides into two daughter nucleus without showing the differentiation of chromosome and development of spindle. Amitosis is commonly seen in prokaryote as well as some cells, specialized cells called nerve cells or neurons. The neurons undergo a mitotic cell division. This means that if the neuron is destroyed, it cannot be replaced because of the neurons do not undergo through mitosis. If we go to the advantages of the amitosis, in this process the cell division is completed within a short time. The amitosis is the major means of reproduction in the lower organism like yeast, bacteria, and some few prokaryotes. If you go to the disadvantages of amitosis, in this process of cell division, there is no possibility of genetic recombination and there is a possibility of expression of unwanted recessive genes. Let me go to second type of cell division, that is endomitosis. Endomitosis is nothing but a type of a cell division where the replication of chromosome in the absence of the cell or nuclear division takes place, resulting in the numerous copies of chromosome within each cell, and it occurs in the salivary gland of Drosophila and other flies. Endomitosis is nothing but the doubling of the chromosome within a cell that does not divide. The doubling may be repeated a number of times in the single nucleus. It occurs mainly in insect where different adult tissues have specific polyploid numbers. It also occurs in some vertebrate tissues too. Dear students, let me go to another concept, cell cycle. The cell cycle is an order series of events involving cell growth and cell division that produces two new daughter cells. The cells on the path to cell division proceed through a series of preciously timed and carefully regulated stages of growth, DNA replication, and division that produces two identical cells or clones. A cell cycle is a series of events that take place in a cell as its own growth and division takes place. The cell then leaves interphase, undergoes mitosis, and completes its division. The resulting cells, known as daughter cell, each enter their own interphase and starts to divide and begin a new round of cell cycle. Let me go to the events occur during the cell cycle. Let me go to the events of cell cycle. Number 1, interphase, which includes G1 phase, S and G2 phase. Second one, phase of mitosis, which also involves prophase, metaphase, anaphase and telophase. Last one, cytokinesis, where it tells about the division of cytoplasm, and then cell will be completely divided into two or more daughter cells. This slide tells about the sequence of the cell cycle, which involves interphase, mitotic phase and cytokinesis. If you go to the interphase, this interphase includes G1 phase where the cell undergo a lot of changes and growth. Second phase is S phase where DNA replication and synthesis takes place. The last one is G2 gap. G is nothing but gap, so where the further growth takes place, and then it is regulated by different kinds of protein and it is proceeded to a next stage, that is phase of mitosis. The mitosis includes prophase, metaphase, anaphase and telophase and followed by cytokinesis. So thus cell cycle takes place. So this is about the sequence of cell cycle takes place in all cells, especially in eukaryotic cell. Let me go to the first stage of interphase, that is G1 phase, which is called as first gap. Because it is called as first gap because from a microscopic aspect the little change is visible. However, during the G1 stage the cell is quite active at the biochemical level. It is characterized by a change in the chromosome from condensed state to more extended state and series of metabolic events that leads to initiation of DNA replication. During G1 phase, the chromatin fibers become slender, less coiled, and fully extended, and more active for transcription. The transcription result in the synthesis of RNAs such as tRNA, mRNA, and rRNA, and series of protein molecules required for the initiation of DNA replication starts here. The length of the G1 phase varies from cell to cell, and also the length of G1 phase is more than other three phases in the cell cycle. G1 phase G1 phase represent 40% of a time of the cell cycle. Let me go for a second phase in the interphase, that is S phase. The S phase is nothing but synthetic phase. The synthesis phase of the interphase is biochemically a phase of active DNA synthesis and histone synthesis. In the S phase, chromosome numbers doubles where the DNA replication and associated proteins are produced. Although some of the histone protein synthesis occurs in G1 phase, most of it is synthesized during S phase. DNA replication is semi-conservative and discontinuous type, which result in the formation of identical pair of DNA molecules. After doubling of chromosomes, sister chromatids are still firmly attached to the centromeric region. At the center of each animal cell, the centrosomes of animal cells are associated with a pair of rod-like subjects or objects, that is centriole, which are at the right angles to each other. Centrioles help to organize the cell division and it is absent in plants. Let me go to the third phase of interphase, that is G2 phase. G2 phase follows 5 phase. This phase represents 10 to 15%, 25% of the generation time of the cell cycle. In G2 phase, the chromosome consists of two chromatids, that is, the cell has twice the amount of DNA content. In the G2 phase, the cell restores its energy stores and synthesize proteins necessary for chromosomal manipulation. Some cell organelles are duplicated and the cytoskeleton is dismantled to provide the resources for the mitotic phase. There may be additional cell growth during G2 phase. The final preparation for the mitotic phase must be completed in this phase, that is G2 phase. Okay, let me go to the mitotic phase. After interphase, the mitotic phase starts. It is the actual cell division takes place in this phase. M phase follows, it is also called as M phase. M phase follows G2 phase. During this phase, the cell divides into two daughter cells with the equal contribution of the chromosome among daughter cells. After M phase, the cell enter into G1 phase and next cell cycle is repeated subsequently. However, cell after completion of mitosis do not enter into the G1 phase. Those cells are referred as G0 cells. The M phase is divided into four subphases. Number one, prophase. During prophase, the nuclear membrane disappears, spindle fibers form, and DNA condense into the chromosome. So, after prophase, the cell will go to metaphase. So, this is the second phase of M phase. During metaphase, the sister chromatids align along the equator of the cell by attaching their centromeres to the spindle fiber. After metaphase, cell will enter into a next stage called anaphase. Ana means separation. Here, the sister chromatids are separated at the centromere and they are pulled towards the opposite poles of the cell by the mitotic spindle fibers. And after anaphase, next comes telophase. During telophase, chromosomes arrive at opposite pole and unwind into thin strands of DNA and spindle fiber disappear and the nuclear membrane reappear. So this is about the M phase, which includes the subphases like prophase, metaphase, anaphase and telophase. This is also called as a karyokinesis. Let me go to the last event of cell cycle, that is cytokinesis. So cytokinesis is the actual splitting of the cell membrane where the cytoplasm starts to divide by the by started by the cell membrane. Especially in plant cell, it forms a type of a plate called cell plate that becomes a new cell wall, especially in plant cell. But in few cells during the cell cycle after telophase, some cell they may enter into a inactive phase, that is called as inactive stage, which is known as a G0 after they exit the cell cycle. Then they are not so active, they are not able to divide, so they will become permanently inactive, especially few cells. In some cells this happens and then there is no cell cycle further. So dear students, this is about cell division and cell cycle, which is coming under unit 2 in second year BSc zoology syllabus, prescribed by University of Mysore and Urajas College, Mysore.